sharing my own. Yes. Okay, so here everyone, thanks for joining the talk, and thanks Kason for organizing this and the nice introduction. I'm Zhen-Qi from Network Neuroscience Lab at the Montreal Neurological Institute. Today I'm going to share with you some recent progress on the understanding functional connectivity. So the talk is mainly based on this paper. It's currently in press at Nature Methods. We just did all the production checklists and something. Meanwhile, you can find the preprint on bioRxiv. We have all the code and data available at our lab's GitHub. So I will start with the assumption that functional connectivity is a very popular concept that everyone has a rough idea of what it is and how it works. So I can skip the basics of data acquisition, pre-processing, parcellation, and all that, and just directly get into the main questions. And I want to start by asking, what is functional connectivity? So most people from my generation probably first learned functional connectivity from some topics in brain networks. The brain is somehow acting as the network, and there is the structural and the functional networks. However, unlike structural connectivity, which usually is some actual physical tracks or pathways, functional connectivity is kind of debatable to some extent, because it does not have the actual physical connections. Rather, we construct the functional networks entirely from statistical estimation. And the most popular way to do it is probably Pearson's correlation. But going back to its basis, it's just trying to capture the relationship between two time series, which does not always have to be correlation. For example, if you are more in the EEG and MEG research, not fMRI, then you're probably more familiar with some of the spectral measures. As you might have known, people have been using many different ways to estimate functional connectivity, and today they're still inventing new ways to do it. And this seminal paper by Stephen Smith is just one of the many attempts to benchmark the many functional connectivity measures people were using at the time. And as a result of these different ways to estimate this relationship, the network in quotes can be very different. There isn't necessarily a single best way to say which one is the optimal one. And also, how do we actually keep track of so many methods? You can see functional connectivity leaves us with more questions and answers. And in this paper, we're going to benchmark functional connectivity using a large number of methods for estimating FC and how they perform in a range of common scenarios. So to approach this benchmarking task, I want to detour a little bit to introduce this kind of new mindset, which is massive feature extraction and profiling. It started with this awesome tool developed by Dr. Ben Fulcher. It's called HCTSA, Highly Comparative Time Series Analysis. In simple words, it's a tool to massively extract time series features. And my first intuition of seeing that is that it would be very useful for machine learning systems. This is true indeed, but it's also more than that. We can, just by calculating the features on functional time series, we're able to find these hidden intrinsic patterns of neurodynamics and link them to potential magnetic candidates. I'll just mention one notable result. The first principal component we found from this is autocorrelation, and the second component is mainly kurtosis. So people used to study single arbitrary features based on their past experience, and now we can survey more than 7,000 features all at once and get insights directly from data, which is quite an improvement in my idea. Okay, so we now know how it works on time series. Coming back to functional connectivity, how does this concept work here? More broadly, I would say functional connectivity is about quantifying the relationship between two time series, and there is a huge amount of literature in the broader scientific fields about estimating pairwise interactions among two random variables. And this is exactly what we're going to do here. So Ben and his group once again brought us another awesome toolbox called PySpy. And instead of time series, this toolbox allows us to calculate hundreds of pairwise interaction statistics. It includes many commonly used measures such as covariance, mutual information, as well as some of the more obscure measures from either information theory or causalities and so on. And we are going to use it for our functional connectivity estimation here. And to give you an idea of what measures we use to calculate functional connectivity, I'm showing here all the pairwise interaction measures we benchmark in this paper. The numbers after these measures are the number of variations belonging to that specific measure. This part is actually showing how they are named in the toolbox. They are pretty readable already. On the left, I have a small reference of a minimap of all their full names. So in total, we use 239 statistics from all these 49 measures. And in the main result, we calculated all of them for 326 unrelated subjects from HCP young adults dataset. Each subject has 4 resting state rounds, and we are using the Shaper 100 node parcellation for this. So we will have this many number of FC matrices for each subject. It's worth mentioning that this is actually not a full list of available measures from the PySpy toolbox. On one hand, they're still updating it. On the other hand, because we're using pretty long time series from the HCP, so the computation is actually kind of forbiddingly long for some of the measures. So we had to remove some of them. This list here is a balance between running time and interesting measures. It still takes a lot of time to run though, but it's more manageable. So with the 239 FC matrices for each subject, we first ask how similar or different are they? And we quantify this by just showing the edgewise similarities between them. So what I'm showing here is the similarity matrix of all the FC measures, with both row and columns being the FC measures. You can see that they are highly organized and form clusters that follows their categories. We also group them or sort them by their categories. For your reference, the conventional Pearson's correlation we commonly use is in the covariance family, which is this top left dark red patch. If we follow it down column-wise, it has a positive correlation with Zinn covariance. We have a negative correlation with precision distance, and then strong positive and negative correlations with some of the information theory measures, and kind of not a lot of correlation with spectral measures. And also the similarities, they are not all at the extremes. A lot of them are actually distributed in the range of -1 to 1, as shown in this bar plot, which means they are not just simply strongly positive or negatively correlated to each other. The spectral measures, for example, they seem to be actually most correlated within themselves and do not seem to be very similar to others. So this could be because we're doing both time series here. But I want to mention that in this paper, although we're doing mostly resting state of mind, it's of course very possible to apply them to EEG or MEG time series. And the spectral measures will probably generate more meaningful potential. So we didn't study them here, but you will see all the approaches in this paper is actually very general and can be easily applied to others if you want. So here I'm showing some of the example FC matrices averaged across individuals. The intuition I want to set here is that they are quite different just by looking at them, just from their appearance. Then it would be interesting, like, would they have different topological and geometrical features? So next we'll start testing some of the properties on them. And we start by showing the distribution of the edge weights. We want to know what are the statistical distribution of the values. The 239 measures are side by side on the x-axis. The darker lines here suggest there is a greater number of edges concentrated. You can see some of the densities are highly skewed. while others are kind of more evenly distributed, suggesting there's substantial differences in topological organization, such as the network hubs. And here I'm showing the weighted degree or the hubness of the measures. While some of the measures, or maybe more than half of them, they have the typical pattern with hubs in mostly dorsal attention, ventral attention, visual, and some of the motor networks, as shown by the darker red here. There is considerable variability across the measures, with some of them having more of a spatially distributed hubs. Some of them even emphasizing transmodal regions. With this in mind, next we check to see to what extent each of the pairwise statistics captures these two very well-studied features of functional networks. The first one we're looking at is the negative correlation with interregional Euclidean distance. And overall, we see that most of these measures are quite moderately correlated with distance. Note that some of them are defined as the dissimilarity or distance between time series, so they appear as positive. That's normal. But there are also some of them having a substantially weak relationship with distance. They're not decaying with the Euclidean distance. The other feature we studied here is the structure-function correspondence. We would expect a relatively strong positive relationship with structural connectivity, and which here is estimated from diffusion MRI. So we can see for most of them this is true, but still there are some of them having a pretty low correspondence. So this too suggests that even for this very well-known phenomenon, depending on your choice of FC measure, it can be this canonical properties can be very different. And after looking at these topological features, we next try to consider how they would be more directly reflecting the underlying biology. And here we quantify this by studying how different types of FC correspond to the other biologically meaningful interregional networks. In specific, we have correlated gene expression from Allen human brain atlas. We have laminar similarity from big brain cell staining data. We have neurotransmitter receptor similarity from multiple PET tracers. We also have electrophysiological connectivity, which is from the resonance MAC data from HCP. And finally, the metabolic connectivity from dynamic FDG PET. So we are asking how well each FC matrix aligns with these networks estimated at different spatial and temporal scales. So in general, we observe the strongest correspondence with this neurotransmitter receptor similarity and electrophysiological connectivity. And perhaps counterintuitively, the correlation with metabolic connectivity here is not as strong in this case. We also find that across the FC measures, covariance, precision measures, they're pretty good in general in terms of correspondence, but things can get quite different for some information theory and frequency measures. So after looking at this more of a group level results, we also turn to some individual level features. We first test how good they can be used to identify individuals, which is a popular analysis also known as fingerprinting. We calculate the identifiability index and found that for precision, or which partial correlation belongs to, is really standing out in this one, which is surprising, but also quite consistent with many previous reports of how this partial correlation might be better at prediction and telling between individuals. And meanwhile, the covariance, where Pearson's correlation is in general pretty good. And fingerprinting might not tell the full picture here. Now we next consider how well these different FC measures can be used for predicting individual differences in cognition and behavior. And for simplicity, we follow these approaches from Ken colleagues. We use this IC-derived 5 components as the target of the prediction. We then use ridge regression to predict component scores for each individual from their each of the FC measures. And we can see here across the cognitive behavioral terms, we generally see greater prediction for cognition and tobacco use, and relatively poor prediction for illicit substance use and mental health. So this magnitude we see here is pretty consistent with previous reports on the prediction. So what we are interested in is across the pairwise statistics, across FC measures. It seems the ones that perform well for individual fingerprinting, they're also performing relatively well for predicting cognition and behavior. So this says it's kind of important to select the appropriate FC measures for a specific task, such as the quantification of individual differences here. So now we've seen how FC measures relate to other types of interregional matrices and external phenotypes. We turn to FC themselves and ask whether we could provide some mechanistic explanation. So we try to do this with an information theory framework, which is kind of complex, but I'll try to explain in simple terms. So back to where we wanted to quantify the relationship between two time series, we can think of them as two blobs holding the information they contain. And how do we study these two blobs of information? Well, one way is to check their overlap, getting kind of the common parts and their unique part. This is kind of the idea behind why people use mutual information or transfer entropy, these information theory measures, as the quantification of functional connectivity. But the framework we are using is actually more general than that. With this framework, we can decompose the information between these two variables into 4 information atoms. As I'm showing here, the 4 colors in this Venn-diagram are on the left. A more intuitive demonstration is on the right. The 4 atoms are unique 1, unique 2, synergy, and redundancy. So the unique ones are pretty easy to understand. It's just what they have that are unique to themselves, not contained in the other party. What's more interesting are actually the other 2 atoms. So synergy here means when 2 sources of information, when they are considered together, they provide some information that is new and cannot be retrieved from either source individually. So I'm using the kind of the yin-yang pattern here to illustrate this. And in contrast, redundancy means each source, they have something exact same information as the other. So when they are combined, they would be kind of repetitive. The framework for getting this information atoms is known as partial information decomposition, or PID for short. Now what we're going to use here is actually one more step beyond this, because we're more interested in the information flow between the 2 variables, which would actually involve an actual temporal step. So back to the Venn diagram, we can replace the labels with the more complete decomposition here, the 4 atoms. And the simplest way to think about the information flow is that if we give them a past and a future state. Now for each of the 4 atoms, they will also have a past state and a future counterpart. Information transfer happens between the past and future, making them different. Now we have in total 16 possible transition patterns. And that's the information flow pattern we're interested in. So this approach is known as integrated information decomposition, or PHI-ID. The 16 information flow patterns, they can be intuitively understood as many common sense terms as shown on the left, such as storage of information, migration of information, or like decryption and encryption of information. And this is how they are mapped from 4 atoms to 16 atoms, basically pairwise. So the theory guarantees all this to work is quite complex, But there's many interesting results and insights from the synergy, redundancy, and the information flow patterns. And Andrea is an expert in this, and he has some excellent papers. If you want to learn more about this framework, you can also refer to these two original papers about the formal theories behind phi-AD and PI. Now back to functional connectivity. So in short, we calculated the 16 atoms for the functional time series, and they look like this. Actually, if you notice the first one, redundancy to redundancy flow, is actually quite similar to what we would expect from Pearson's correlation, right? While some of other of them are more asymmetric or pair differently. And by the way, here's an open source toolbox you can use to calculate these measures on your own data. And with this information flow atoms, we want to see how our set of FC measures are represented by these information flow patterns. We actually constructed linear models and estimate the contributions of each of the 16 information flow patterns to each of the FC matrix. So the rows are the 16 patterns, and within each column, the darker or lighter say something about their relative contribution. We find that for classical statistics such as covariance, precision, and mutual information here, they mostly reflect the pattern that is the first one, which is where the redundant information stays as redundant. That's why we see the first matrix above is similar to what we would commonly expect from Pearson's correlation, meaning that they are mostly looking at how the repetitive parts staying as the repetitive part. So this is both reassuring, but also kind of worrying in a way, because there's a lot more to study that might be more interesting. So for some of the spectrum statistics, you can see they actually predominantly reflect some unique to unique patterns. A pattern where the information that was exclusively provided by one region stays as exclusive to that region. And both of the cases I described would belong to a pattern of information storage, as I mentioned before, where the information is kept the same over time in the same place, but like there exists much more diverse information flow patterns than this. For example, for some information theory measures or causality measures, or some integrated information, like they were kind of specifically designed for this purpose, we can see either the transfer of synergistic information, which belongs to encryption or decryption, which more formally known as downward and upward causation, if you're more familiar with these terms. So there's a lot going on here, and we actually did not try to interpret each one of them. What we are trying to convey here is that there exists a full spectrum of information flow patterns that are yet to be explored. And this is definitely not possible by just looking at commonly used Pearson's correlation. So I think I'm going pretty fast, and this actually concludes the main results. And I just want to show some sensitivity analysis we did. So what we did is mostly testing the changes for the symmetries between the functional connectivity measures we had. First, we had it within the main dataset. There is a very high correspondence within the dataset, regardless of the different splits. But then we changed parcellation atlas from Shaver to Decimal Canani, and we changed parcellation resolution. It's kind of forbiddenly long to run on any larger parcellations for some of the measures. We also finally tried global signal regression. There's this kind of weird pattern here, but in general, they all seem to be pretty robust. The other thing we tried is to do a correlation with motion. Notably, here we do see a decreased relationship. Seeing this there's this lighter bar here. This is the precision or the partial correlation measures as what we would expect. And finally, at the request of reviewers, we added more data sets to study from just HCP adults to a bunch of more data sets. And in general, we find pretty correspondence between the different data sets. I'm only here showing the group level results, which is the similarity matrix in the first figure, this one, and how they look like on the other 5 data sets. And this is how they correlate with each other, right? Well, I have more results of individual differences between the data sets in supplementary materials in the upcoming paper. So the reviewer also thinks HCP is kind of special in terms of the acquisition protocol and asks us how this might influence our results. It usually be quite hard to test, actually, but luckily, TestLab has this amazing resource called reproducible brain charts. I have a link here, and I think they have a preprint company. Where they have the NKI dataset fully pre-processed. It has 3 different sequences and processed with 2 different denoising methods, which makes us able to compare the FC matrices across sequences in a controlled setting. You will see that across the combinations of these 6 cases, they were in general quite robust. It's a bit different from the similarity matrix we saw before, but in general, they are pretty similar. So finally, I want to summarize this paper by providing some conclusions and recommendations. So in this paper, we see that different statistics give very different FC matrices, and there's the substantial variability, even for very well-known phenomena. This means FC measure is quite depending on the context. And this also alerts us that there is not necessarily a single best measure, but rather be like different measures targeting different mechanisms. And without a ground truth, it can be tricky and important to pick an FC measure. And that would depend a lot on the research question at hand. So some of you might be interested in like, what exactly FC measure should I use in my research or kind of like that? Well, we do have some general recommendations, and I kind of don't want to go like into too detailed to like actually rank them. We do have a ranked list of all the tests in the supplementary materials. But here I'm just providing some like kind of general ideas. So we say that FC measures should be matched to the experimental questions you have in mind. We do see that covariance, distance, or the Pearson's correlation is in general pretty robust. So there is a reason that they are used so commonly. We also find precision or partial correlation measures. They have some desirable standing out features in some scenarios, such as in predicting individual differences. And finally, maybe it's possible to combine or engineer multiple FC measures together for some specific sensitivity or precision purposes you want to test on. And as for future directions, I think first, as you can see, there exists a lot of biological complexity in measuring the FC. Different modalities, FC measures, they may capture different aspects of it. And there's definitely a lot more to explore, both theoretically and experimentally. And I really do want to recommend this review paper about advancing FC from association to causation, about understanding more behind it. The fact that we are seeing partial correlation standing out in some cases is a great example, which is also highlighted in this paper. So in case you're more interested about massive profiling and things we did not cover, I want to call out to 2 other excellent work, both using the PySpy toolbox. One is from Annie Bryant and Ben's group. The paper is more about machine learning prediction in this case. Another one is from last year's OHPM abstract from Conor Lane and Greg Kerr, where they tested a number of different pre-processing choices. Okay, so I think that's kind of all for today. I'd like to thank my lab mates, collaborators, and thank you again, Kaizen, for organizing this. And here you can see like how to get in touch with me if you have any questions and where to find research updates from lab. I think I'm going pretty fast, so I can take some questions.